on your independent research project. But because we do have a lot of people that are new to this, I want to start off by just kind of briefly, and I will try to really keep it short, but kind of go over the major parts of the program, how it is organized, and then I will pass it on to Sonalika and Dr. Harpreet Kaur to guide the session for today's presentations. So let me start by kind of introducing you to our background, who we are. So as you all know, OmicsLogic is a bioinformatics training portal that has been developed by this collaboration that we have between Pine Biotech and the Talbot Bioinformatics Research Center, which is a research center at the University of Haifa. But over the years, we have had the chance to work with students, researchers, and clinicians from many different institutions from around the world. And as a result, we now have this global community that has over 10,000 participants from all around the world. And we have gone through many steps of iteration to make sure that our program is structured and well prepared for the needs of those that are interested in bioinformatics and data science in application to a variety of different biological and biomedical challenges. So we have a dedicated team in India. We also have a number of pilots that we have done at different universities. And our background is that we have done a lot of in-person workshops as well as different types of online programs that we've conducted in the past couple of years. We also have a dedicated team in Africa that is spearheaded by our team in Nigeria. And we also have a number of other participants from other different countries. And we have been trying to work with the local and regional partners that we have to make sure that our content and materials are well adapted to the needs of these local participants. And so we think that we've gotten very positive feedback, and we've conducted these again in India, in Africa, and obviously also in the US. So in the US, our major partner is the Louisiana Biomedical Research Network, but also Georgetown University and others. And so what this allows us to do is to really bring you the best material from all of these locations, consolidated and adapted to an online experience, leveraging our experience offline and kind of trying to facilitate this process of research and discovery using bioinformatics. And so the outcome that we see as a result of this kind of training is the ability to guide and help complete research projects conducted by faculty, teams of people, as well as individuals. So the research fellowship combines access to big data, which means big data resources, as well as tools to analyze and process big data, mentorship, which is both experts, trainers, and peer example, as well as access to a variety of training resources for asynchronous and live training. And so what we are trying to address here is the problem of helping people access resources for big data analysis, mentors that can help match the right kind of analysis methods to their research question, and a community that will keep you accountable and provide you with feedback so that you are prepared to work on impactful topics and develop your research projects until the very end when you are ready to present. And so this comes with a lot of program resources that we've developed that we think could be very helpful for all of you. Now, we tried to combine all of these resources on our OmicsLogic training portal that has a variety of different types of asynchronous courses, as well as more guided programs that are led by experts in bioinformatics. And we provide for all of the participants of this program access to specific sessions that you can access to understand whether that program is right for you. And this is typically done in collaboration with other universities and organizations. So not only do you get the opportunity to learn about the topic, but also interact with participants from these different institutions. But you can find all of this on our OmicsLogic portal, which is learn.omicslogic.com. You will find there adapted courses for you personally based on your experience as you progress. That will change You will be able to track your progress and see how you've improved your understanding of the basic topics, as well as the application of these different tools in different types of challenges and projects. Like this is an example of a project on the origin and pathogenesis of the novel coronavirus, and we have many other examples like that as well. So behind this, what we also provide is access to user-friendly big data analysis tools. And this is a critical component of this program. You get access to 5 terabytes of storage, which allows you to process and analyze large data sets that allow you to use advanced statistical methods and machine learning, as well as multi-omics integration, and several tools that will be helpful for biological interpretation. Now, this infrastructure is limited. We do not give everybody access to that same resource, and the research fellows that have joined this program have that kind of a special level of access where you can actually conduct a full research project, but you have to coordinate it with us. And so what that allows you to do is to explore a data set and find the right methods to apply to a research question. So the objective of this research fellowship that I will briefly discuss today is to start from training and then guide you to complete some of the training resources that I've identified, create a sense of understanding and practical skills, and then to apply that to a number of research problems. Now, the best way to get started is to really take a look at a variety of projects that we already have available. And these range from topics in biomedical applications to biotechnology to a variety of human health related topics, as well as more advanced topics in cancer, biotechnology, agriculture, neurodegeneration, infectious diseases, and ultimately any other topic that you would like to focus on. But it's important to come to that topic prepared, to really be able to understand how experts work on projects, to understand how to find data sets, to specifically identify a research question, and then find the right tools. And so the research fellowship is really an in silico research experience where you have 3 to 6 months, typically. Some of you I know have joined for only 2 months, but typically it's going to be from 3 to 6 months. And you're able to work as a part of a structured program. And today's session is one of these examples where you hopefully come away with a clear understanding of what the program does and how it's organized so that you can navigate the different resources with ease. So a critical point of that is this structure that we've developed that is divided up into 3 parts. First, there's the introduction, where you get the introduction to bioinformatics theory, methodology, and applications. You get to use some of the cloud tools for big data processing and also complete some of the introductory coursework. Then it's the evaluation, where you start to analyze literature from other publications and other data sets to understand how to develop a research proposal. And then once you have done that, you can apply the tools that you've learned how to use to a problem of your own. To help guide you through that process, we have 2 specific channels that help us help you. One of those is our Slack channel. So at this point, can I ask everyone that's present today, how many of you that are actually a part of the research fellowship have joined the Slack channel? Okay, if you have joined the Slack channel, please put in the chat yes or 1 or something like that. Okay, thank you. All right, excellent. Okay, thank you, everyone. So if you have not gotten access to the Slack channel, I see Nandita, Aishwarya. If you have already applied for the research fellowship and received your acceptance letter, you should be added to the Slack channel. So please reach out to marketing@PineBio, and Sonalika, who is here today with us, will be able to add you there. The second important part, and Sonalika, maybe you could be in contact with the people that said no to make sure that if they have applied and gotten their access, they should be a part of that.
Okay, so I would want to add here that I have sent you all the invitation already for the Slack channel. Please check your mail IDs and then you'll get your invitation there and then you can join. I'll send it out again. Thank you.
Thank you. And the second one is this weekly progress report form. So once you are a part of the research fellowship and you would like to discuss any updates, technical challenges, or questions that you might have, before a weekly session, you have to submit the weekly progress report form. Okay, so I've seen a few of you have submitted that form today. Typically, you would want to submit that form a few days before the session so that our team can prepare and address the specific questions that you have, or if you have a presentation that you want to share your progress report with us, we can help you address any kind of like immediate questions. And so I will talk a little bit about the presentation here in just a second as well. The second part is access to these publicly available data sets. So when you collect raw data for your project, you have to be familiar with what this data represents. Okay, so a big mistake that many people make is the first data set that they come across, they start working on that data set. Before you start analyzing anything, the reason why we ask you to submit this progress report is that we want to know that you are trained and ready to analyze a particular type of data, and also that you have explored the data metadata. So how do you explore the metadata? Typically, that means you have to read the paper. And in the paper, you will find that associated data set, but in the paper, they also describe how did they generate the data set? Why did they generate it? What were the questions that they were asking? And that typically results in a specific design that helps you extract from there the types of comparisons and contrasts that you would be able to do in your own research project. So typically, you also have to perform exploratory data analysis so that you can understand the variability in the data set. And oftentimes people will end up using multiple data sets. So you have to be familiar with methods to normalize your data, explore your data, address any kind of technical variation, and only then ask that specific question. So hopefully the tools that we provide will be sufficient for you to be able to do that quickly. Now, after that, not before that, but after that, you choose a topic, right? And in that topic, you want to be very specific and find a topic that is compelling and interesting to the audience that you will present, right? So you have to have the data set available, you have to understand what is the background, and then you also have to do literature review to understand where is that specific topic that you would like to focus on for the next 3 to 6 months. If you do that before you've explored the data, before you've read the literature, you're not going to be able to find an impactful topic that you can actually work on. So here we provide specific training, and I'm not going to do this today, but you've probably seen this in previous sessions, and if you have not, you can request that recording, or you can also join one of our upcoming sessions where we will do this kind of training, where we go through how to perform literature review, how to identify a specific topic of interest, how to ask a research question, and ultimately how to translate that into a particular question or hypothesis that you can work on. And that's where your mentor really comes in. So as a result of analyzing your activity on the OmicsLogic portal, and as a result of looking at the specific coursework that you have completed, the articles that you have submitted in the research proposal, and other things, our mentor will be able to use that time effectively. So we don't want to use the mentor time for just going through the very basics, how to open Excel, how to install R, how to find a data set, things like that. That's where these weekly meetings are for. The mentor session is to help you address the specific question that you might have and make it more impactful. And so for that, we've developed this step-by-step process where you identify a topic, you find the data set, you make a table of metadata and you explore your data. And then when you conduct your analysis, you put all of that into a shared presentation. And that presentation should be shared with your mentor. That presentation is where we can find all of the updates that you're making and see how you're making progress so that we can help get you unstuck in technical issues and also point your attention on the most interesting aspects of your data set. When you present to the mentor, you're not only presenting to the mentor, you're also presenting to the peer community that is all a part of this research fellowship. So today we will hear a number of these project presentations. We will give feedback, but also we ask that you ask questions, you pay attention to the presentation, and you think about how is this project addressing the specific need or question that was proposed at the beginning of that question, right? So that process of information exchange and participation is also a critical part of this program. Once you incorporate all of that feedback, then you can finalize the presentation and potentially identify a poster presentation or a research journal for peer review that you would like to submit. And that's something that we will talk about today, how to make your presentation impactful and organized so that it is making sense and it is effective at conveying the story. When you process the data for analysis, it's important to understand that some of these data sets are huge. And when you go through these data sets, you have to follow a specific process. And so that's where the training is very important. But beyond getting the basic data set organized and prepared, you then have to be able to explore the data. And that's why we also provide you with training on how to use basic tools like Excel, R, Python, and different online tools that you can leverage to explore the data set that you have selected and make sense of it. So please leverage some of the tutorials that we have on the learnomixlogic.com portal, and that will help you deal with that data set. Once you perform the in-depth analysis, we have several experts that can help in a variety of different topics. For transcriptomic data, for structural data, for genomic data, for different parts, we can match you with somebody that can help answer your specific questions. You have to let us know. So remember that it's your responsibility to reach out to us and let us know what is it that you're interested in doing and how we can best help you. Finally, when you get to this process of preparing your analysis results, you are going to be also following examples of many others that have gone through the same process. And so that's where your peer community is going to be very helpful. So when you present and you go through these iterations of drafts, you will see that we can help you the most by making that presentation better and helping you present something to external evaluators at poster presentations, if you're looking to submit a research article, in a much more prepared way. And so the outcomes of this process are very important to keep in mind, right? So have a goal when you're joining this program, have a specific goal. You will have a guarantee and a positive outcome if you properly identify what that outcome for you is. Maybe you just want to understand the specific challenges and data sets and gain training. That's one option. Maybe you want to develop some skills like processing data and analyzing data. Maybe you want to join an independent project or a group project. So that's another option. You don't have to work on this on your own. We will hear today from Rutvi, who has completed an independent project, and hopefully she will continue to work on a group project next. So some people will actually announce that I'm working on this topic and I'm looking for collaborators. That's why it's important to be a part of the Slack channel and our community so that everybody can get to know each other, not only hear their presentations and the final results. So let me briefly tell you what are some of the recent examples of people that have successfully completed the research projects and the research fellowship. April Zhang just recently completed a research fellowship. She's a high school student, and now she got accepted to Berkeley for bioinformatics undergraduate. And we have a recording of her presentation that you can take a look at. I'm going to post the link right here. Simai Dolaner, she will be presenting in the next Psyomics session. That will happen, I believe, tomorrow, but I will let Rutvi talk about that in a little bit. Supantha, who is from Bangladesh, and he is also, you can see his project presentation. He will also be presenting at the Psyomics Club. Dr. Anita Gansa, who is right now working still on a number of research projects together with her team. And we have collaborated with her and her team to recently present her project at the Louisiana Biomedical Research Network. So please take a look at some of these links and you'll kind of see how this works for some of the other people that you might be following in their footsteps. And all of them are, of course, going to be available as somebody that can give you hints and tips and maybe some guidance on how to complete a project and make it impactful. So that's an important part of the outcome as well. When you think of completing this project, think about how are you going to use it to also advance your personal career and learning. And so we have an example of Urja that finished her project a while back. Andres, who has completed his project and will be presenting next week. We have others that are selected to be a part of this program. And so please do share that information with others, with your network, to help them understand what you are working on. So now let me pass this on to Dr. Harpreet or Sonalika. I don't know who is going to announce who is going to be presenting next, so that you can kind of see where this process will lead you as you join this research fellowship.
Thank you so much, Ilya. I would want our new research fellows to introduce themselves before we start with the presentations. So Arnold, can you please introduce yourself? We have Arnold Jean with us today. He has just joined our program. Arnold, over to you, please.
Yes. Hi. Thank you, Sonalika. I'm Jean Arnaud, as you see in the picture, except the name's backwards. Coming from originally from Haiti in the Caribbean, but now in Massachusetts. I was trained as a chemist, but and I worked in biomedical science at an institution next to MIT. But while working there, I realized I was mostly focused on metabolomics, so small metabolites that or biomarkers for diseases. But one thing that became very evident is that so here I am working with the LCMS and other mass spectrometry instruments, and it became very evident that a lot of biomedical research is actually systems biology, and I had no training in R or Python or metagenomics, and so I looked for opportunity to develop some skills, and I did find a few training opportunities, but I always felt as though it was not as well organized as I would like. So when I discovered this program, and I had the chance to talk to Ilya Brodsky, one thing I said was that, amazing, you guys are actually giving me the logic of all of these tidbits. And so I signed up recently, and I'm looking forward to learning more. Last thing I'll say is my particular interest as of right now as a topic is long COVID. I have a few friend of mine who had COVID, recovered, but there's continuing symptoms after you've had COVID, and that is referred to as long COVID. And so I'm very much interested into some of the bioinformatics associated with that. Thank you.
It was great to know about you, John Arnott. That's how you spell your name? Okay. And now I would want Tanmay to introduce himself, please. Tanya Arora.
All right. Thank you so much, Sonalika, and thank you so much, Arnold. I'm really fascinated by the kind of research that you're into. So my name is Tanya Arora, as you can probably see on your screens right now. And I'm currently working as a researcher at different labs and on a few different R&D projects. So I'm currently working at the Indian Institute of Technology in Gandhinagar. And I'm also working at IIT Delhi and the Defense Research and Development Organization in New Delhi. And one of the things that have always fascinated me is the integration between computers and cells. So if you think about it, computers and cells are essentially the same at the most fundamental level. They're both really complex machines, and they happen to share very similar concepts of signals, networks, pathways, and even memory. So it was this curiosity towards computers and cells that really got me into research in the first place. And over the course of the past 5 years, I've employed all of my time and my resources towards the large goal of deconvoluting the very wiring of cells and slowly trying to understand how life in itself is digital in nature. And I believe that's really the primary goal that we're looking for out here at Pine Biotech. And in addition to that, I'm currently working on the topic of Nano Optogenetics. So the project that I'm working at IIT Gandhinagar, it's centralized around the very viability of incorporating different stimulus responsive proteins in optogenetics and the potential benefits and challenges of this novel focal stimulation approach inside of the whole entire process of nano optogenetics. So that's about me.
Thank you so much, Tanmay. Now we'll take up the other introductions maybe after this presentation. So now I'm going to hand over the stage to Ruthvi, who would be presenting on a project. Ruthvi has already completed her project. So let's see what she has done throughout her project and learn more about it. Over to you, Ruthvi.
Yes, can you see my screen?
Yes, we can see.
Okay. Hello everyone, I'm Ruthvi and I did this internship with Pine Biotech and I worked on the topic of multiple sclerosis. I was very fascinated by this topic and the kind of different characteristics it shows, like lesions and everything we will look ahead of. So the reason for choosing this topic was this one. So I'm an undergraduate third year student of biomedical sciences and I'm from India, Gujarat. My interests particularly are neuroscience, cancer neurobiology, and bioinformatics, precisely neuroinformatics. I've worked on DIPG, multiple sclerosis for this project, and autism and immunology. So before diving into the project, I would like to brief you all about what actually multiple sclerosis is. So it is a central nervous system disorder, mainly a white matter diseases, which is a neurodegenerative diseases and it is characterized by the demyelination of the myelin sheath in the axons. Because of demyelination, the patients of multiple sclerosis suffer an acute episode of neurological disturbances. And also due to this demyelination, the balance between the brain and the body, that is the normal rhythm, also gets disrupted. So usually multiple sclerosis till now has been regarded as a white matter diseases, but since last 5 years, there has been extensive research that shows that not only white matter, but multiple sclerosis also affects the gray matter. Still, the underlying causes of it have yet to be unfolded. So talking about the morphology of multiple sclerosis, the pathology is characterized by the multiple focal lesions and the MS plaques that develops in white matter as well as gray matter areas of the cortical regions of the brain. So till now, scientists have found out that T cells, that is activated T cells in the lymph nodes, enter the CNS through blood-brain barrier, and then they start attacking the myelin sheath of the axon, due to which demyelination happens. But in the first place, there has been no research till date which shows that why these T cells or say B cells get activated and attack the myelin sheath. So talking about my research question and hypothesis, so my main research question was that patients of multiple sclerosis have less or deemed cognitive functions, and they usually tend to forget things and cannot pay attention to any particular thing. So memory loss is one of the most important characteristics of MS. Also, so using this main research question, I wanted to answer my hypotheses that were 3, divided as first. I wanted to know the biological pathways and their respective functions that were affected in patients that had gray matter lesions, that is MS patients. I also wanted to find out that does multiple sclerosis precisely have effect over the development or differentiation of pro-T cells, because you will see ahead that I had gene ontology results presenting hematopoietic stem cell lineage pathways as well. And I also wanted to know that are there any specific genes in multiple sclerotic lesions that they are upregulated or downregulated, and any specific role with any biological pathway that they play or not in multiple sclerosis. So owing to this hypothesis, I started searching my data set. So this was the data set that I found, and it consists of tissue samples of cortical tissues, and it had total 184 samples in totals, out of which this is a super series. So I took only one sub series that had 78 samples, which had gray matter lesions versus control gray matter. So this is a slide depicting my study methodology. So firstly, what I did was RNA sequencing analysis. So I had three types of sub data sets, like the sub data experimental groups. So first one was the gray matter lesions. The second one was normal appearing gray matter, and the third were controls. So firstly, I wanted to explore all the conditions by comparing them with each other. So first I did my exploratory analysis, in which I compared all the three of the groups with each other by doing a t-test under all conditions, that is all three. Later on, I compared each two of them with each other, that is lesions with normal appearing gray matter. By normal appearing gray matter, I mean that that person has multiple sclerosis but do not have lesions. So I compared lesions with normal appearing gray matter by PCA, and also compared controls with normal appearing gray matter. The variation I saw was good in both, but the most variation I could see was among control and gray matter lesions, which we will see further. So I downstreamed my analysis and then went for comparative analysis between gray matter lesions and control gray matter. For differential gene expression, I used EDGE-R, and after getting upregulated and downregulated and significant genes, I did gene selection with top 700 genes that I got. And further on, I did pathway analysis. Again, I did visualization by PCA and then heat map for all the significant genes that I had extracted out. And later on, when I got my results, I did literature review in order to compare my results with previous studies and get conclusions and derive conclusions from them. So talking about my exploratory analysis, so this was the first PCA I did. On my left-hand side, you can see the PCA between normal appearing gray matter and gray matter lesions. And on the right-hand side, you can see controls and gray matter lesions. So you can see that after downstreaming my analysis, the variation among this, that is healthy and gray matter lesion, was very well. So I took forward my downstream analysis with this part of the data set. This is a slide depicting the heat map with significant genes. So I had selected significant genes with p-value less than or equal to 0.05. So in this heat map, it can be clearly seen that most of the significant genes are downregulated in the lesions, whereas are downregulated in the control samples, whereas most of them are upregulated in gray matter lesions, which showed a clear indication that in lesions, something is upregulated, which leads the formation of inflammation, or rather say lesions. So after selecting upregulated and downregulated genes and my significant genes, I did pathway analysis using the human gauge pipeline on tBioInfo server. So the top two heat pathways that I got was, first is olfactory transduction pathway, and second I got was hematopoietic stem cell lineage pathway. So for olfactory transduction pathway, the R1 you can see over here, that is the olfactory receptor R protein, which is responsible for sensing the odor and smell detecting pathways, was highly upregulated in gray matter lesions. So I'm sorry, downregulated in gray matter lesions, due to which it was a possible association that people who are detected with multiple sclerosis have a lowered sense of smelling and odor detection. The second pathway that I got was hematopoietic stem cell lineages, and it could be seen that among the whole picture of this pathway, interleukin-3 is highly downregulated. So over here, association can be made that is downregulation of interleukin-3 a possible reason for T cells attacking the myelin sheath, or underdeveloped bone marrow a possible cause of multiple sclerosis onset. So after this pathway analysis, I did pathway analysis with enricher with particular downregulated genes. So in downregulated genes, you can see that the top hits were all olfactory receptor activities and perception of smell pathways, which clearly indicated that this pathway was disturbed in the patients of multiple sclerosis. Further, I did pathway analysis with upregulated genes, which showed top hit in ribosomal protein synthesis and oxidative phosphorylation, which were also the results I got on HumanGauge TBio info platform server pipeline. So after that, I had to make associations. So the possible association I could make was the downregulation of olfactory receptor proteins and multiple sclerosis. So what I did was, out of the significant genes expressed, I picked out the genes which were responsible for the olfaction, that is OR genes, and I plotted a bar plot comparing it with GML lesions and control gray matter. So in the bar plot, it is quite evident that in lesions, these genes are downregulated and as compared to the control. So these were the top 12 genes. Again, this was my hypothesis. So I needed my validation to be concluded in order to prove that my findings are true, which would come ahead in the next slides. So after that, I also found that protein kinase A, which was severally related to my studies. So protein kinase A generally functions in cognitive functioning, like in activation of brain reward system or in preservance of short-term and long-term memory. So as per our KEGG analysis, PKA protein in the odor detection pathway is highly downregulated in multiple sclerosis patients which had lesions. So we can also make a possible association. I'm not saying that I'm quite sure, but over here I'm talking about an association between PKA and the loss of cognitive functions or the memory loss in patients of multiple sclerosis, because over here in the picture it is quite clearly visible that protein kinase A is present in nucleus accumbens, and protein kinase A is directly activated by dopamine receptors, which are responsible for producing dopamine. A loss of dopamine is also an important characteristic in patients of multiple sclerosis, and hence a possible relation can be made between PKA protein and memory loss in multiple sclerosis. Moving to the analysis of hematopoietic stem cell lineage pathways, there were 3 or 4 possible questions and associations I could possibly make. So still more research is needed in future to assess the precise effects of multiple sclerosis on differentiation of protein cells. But however, downregulation of interleukin in the whole hematopoietic stem cell lineage pathway shows that thrombocytopenia and neutropenia, which are often the main characteristics of patients of MS, regulation. So talking about the hypothesis, that is the top 12 olfactory genes that we found that were downregulated in GML patients, that is MS patients, I took another data set that had 106 samples. So 106 samples were divided into cortical normal appearing gray matter versus control gray matter. So again, this was a brain cortical tissue, gray matter tissue sample and I used this as a different data set, and I used this for validation. So to my excitement for the validation, there were also other olfactory genes that were downregulated, but I extracted the 12 genes that were there in my hypothesis and compared it to the controls. And in validation, I exactly got the same results. That is, these genes were as well downregulated in multiple sclerosis patients without lesions as well. So hence, it was pretty sure that olfactory dysfunction and olfactory disruption pathway is seen in patients of multiple sclerosis. Also, for a better picture, I have plotted a box plot representing the downregulation of genes in normal appearing gray matter, the olfactory genes, and they are normally expressed in healthy controls. So my key findings and suggestions include the top 12 olfactory genes, which were shown to be downregulated in multiple sclerotic patient, a possible association between protein kinase A, cognitive loss of functions, and compared to multiple sclerosis. Also, as per from our KEGG analysis, a possible association can be made between suppressed production of interleukin-3 and thrombocytopenia in multiple sclerosis. For my future directions, I would state that much research is needed in this area to assess the precise effect of multiple sclerosis on protein cells differentiation and development. Also, more comprehensive research is needed to support my findings. And I'm done with my presentation. Thank you so much.
Thank you so much, Ruthvi, for sharing with us your research work, and it's really great. Now, I would want our mentors to please provide the reviews and the suggestions to Ruthvi.
I mean, I just want to say that, Rudvi, it's very exciting to see your presentation kind of coming together. If you could please go back to your first slide, just so that we have something in front of us. So I think that, you know, it's very interesting how you went through this project and kind of developed your hypothesis to then finding the specific signal, then focusing on validation, and you ended up with multiple questions at the end that I think do require further research. So I really like the way that you structured the presentation. And I'm hoping that, you know, as you move forward, I know that you still have some time left, that you will find an opportunity to collaborate with other people on addressing some of these questions, perhaps, or if you have some, you know, kind of further questions that appear that you would like to focus on. So could you speak a little bit about how this process worked for you? You know, how long did it take? When did you kind of clearly identify that specific question that you had in mind? Like, what was that like for you?
Okay, so to be honest, I started in mid of February, and I am a biomedical sciences student with a zero experience in bioinformatics, and I'm not at all ashamed to say this, that I did not even knew that what NCBI stands for, or is there even a website named NCBI. So firstly, I had to do all these courses, and after doing courses, I didn't even knew what data sets meant. So I had few sessions with Harpreet ma'am. So she taught me about how to find a data set, how to extract that. So everything and finding a correct data set took me 2 months, to be honest, finding it on my own. But after that, doing PCA, differential gene expression, I obviously had technological and technical problems as well. But you know, when you learn on your own after doing a thing, you text your teachers and they give you a time, but you cannot wait in between for the specific amount of time. So you start reviewing articles or searching on YouTube the videos that the institution has uploaded in order to solve your query on your own. So one thing I learned in this project is to overcome difficulties on our own and then going to the mentors, because mentors have really an expertized way of selling them. And after talking to my mentors, everything seemed so easy. I remember in February, there was a time when I didn't knew anything and I had to, you know, exactly ask my mentors, what should I do? What is PCA and everything. But after 4 months, now I am helping my fellow colleagues and the persons who are working with me. So it has been an incredible curvy journey, but I have definitely learned a lot from this.
Right, great. Thank you very much. I think that's a great perspective. So thank you for sharing again. I know it's been a long journey and really excited to see the results.
Sure, thank you.
Dr. Harpreet, would you like to add?
Yeah, hi Ruthvi, and it is very nice presentation and I'm really happy to see your progress. And I have just few questions and maybe you just need to, or maybe you need to correct them. So first question regarding your, where you are identifying significant genes. If I do remember, we have not taken p-value, that is p-adjusted value. And as well as we also consider log to full chains, and please, I mean, correct that on your slide. So another thing that is when you are working on significant genes, I mean, especially on the validation data, and you are drawing bar plot. I think in comparison to the bar plot, even it's much better, maybe heat map. So and another thing, can you just go to your last slide, second last slide, where you're trying to show the results? I mean box plot type.
This one?
Yeah, I think it is somehow not, maybe you just need to replot it. It is not clear. So I can show you like how you can do it in R. So yeah, this is something that you need to, I mean, need to redo.
Sure, ma'am, I'll make these corrections and let you know.
Okay.
Okay, thank you.
Thank you, Ruthvi.
Thank you.
Thank you so much, Dr. Harpreet, for your reviews. Now I would like to invite Asma to share her presentation. Over to you, Asma.
Thank you.
I'm making you the co-host. You can share your screen then.
Okay.
Yes, please go ahead.
Yeah. Can you see it right?
Yes, we can see.
Okay. Welcome to my review session. I am Asma Atılgan from Turkey, Bilkent University. My research interests are epigenetics, immunology, and autoimmune diseases. I completed 3 courses: genomics, transcriptomics, and epigenetics 1, 2, and 3. I select the disease which is called rheumatoid arthritis. It's an autoimmune disease. It can develop at any age and it can affect women more than men. Many genetic and environmental factors appear to be involved in this disease. And when we look at its treatment, disease-modifying anti-rheumatic drugs are used. This disease has some stages which are early, moderate, severe, and end stage. Also, there is a stage called remission, and it's the total absence of symptoms. I select these data sets to further analysis. This is an RNA-seq study of synovial biopsies. It's an expression profiling by high throughput sequencing. I look at the disease states in these data sets, which I'm interested in, especially normal, healthy people, established rheumatoid arthritis patients, and early rheumatoid arthritis patients. What did I do from now on? I select my data sets. I did principal component analysis. Then I used DSEQ2 for differential gene expression analysis. And then I did enrich-R analysis to look at the gene ontology and cake pathways. And I want to show them to you today. And these are my DSEQ analysis results. This is for the early and established rheumatoid arthritis patients. When I look at the pathways, the nicotine addiction drew my attention because I know that smoking increases the risk of rheumatoid arthritis disease. And also some other pathways are important. And I look at these go biological process functions, etc. And this is for the normal and early rheumatoid arthritis patients. And these are my results. Actually, I want to ask, what should I do for the next steps when I looking at these results?
Hi, Asma.
Hi.
So I mean, here you have just like performed analysis. Now you need to explore like what are these pathways, right? So
Yeah.
I mean, you need to do analysis on this, like how these pathways biologically affected with the rheumatoid arthritis. So, and another thing, can you just go back to your previous slide?
Which one?
Like how, I mean, how many upregulated and how many downregulated genes that you have identified? I mean, you should consider those things whenever you're presenting your work, like maybe in terms of table or something like. And then now you, most important thing, you need to analyze like which pathway, so how they biologically, I mean, affected this arthritis, arthritis disease. So you need to, I mean, you need to correlate these things maybe with the literature as well, and you need to understand how these pathways actually affect the disease, right? So that is something that you need to explore. Yeah.
Okay. Then I will look at them. And because
I mean, how many you need to identify, and also need to mention how many genes that you have identified here.
Yeah, I forgot to write it. Sorry.
Yeah, I mean, you need to explore these kind of things. So yeah, and I would like to ask to Ilya to add, I mean, suggestion.
Yeah, thank you. So I think that, you are starting to put together this project and you got to a point where you find these differentially expressed genes. The differential expression is supposed to tell you what are the biological differences. And one of the challenges is to take many different sets of genes that are grouped together by pathway, like you're showing here, and to make sense of how this is interconnected. So as you've seen Rutvi, she has used an explanation of what the pathway is overall to show us how different genes are influenced by this condition. She also wanted to get at the end to a set of genes that she could validate, right? And so I think in your project, it is a little bit of a similar approach. You describe the problem statement, you then define, basically when you're doing differential gene expression is, that's a contrast that you've selected to do. And so even before that, I would add some slides on PCA or kind of a summary, a statistical summary of what it is that you found overall in your project.
Actually, I was adding, but as I have a limited time, I skipped them.
Yeah, this is fine, but this is an important actually part of your presentation because maybe, here we see technical outliers rather than, maybe you've addressed that, right? So that process of defining what the data set looks like to be able to ask the differential expression kind of a test, right, is something that you want to present to us as well. And then when you finalize your gene expression analysis, show us what do you think the, genes mean. Okay, so maybe in your slide 22, I don't know, what is slide 22?
This one?
Mm-hmm. So like here, it seems like you have a very significant difference, and that is typically defined by these parameters, the adjusted p-value, the log fold change, and so you can modify them to focus on, significantly expressed as well as significantly differentiated, maybe making sure that you're selecting the right kinds of genes for downstream analysis. And so what I would say is that at this stage, your presentation is a good time to schedule a one-on-one meeting with Dr. Harpreet to kind of discuss more in depth what are all of the findings, to sort through them and focus on some specific patterns that you are finding.
Okay, thank you very much.
Thank you so much, Aisman. Now I would want to invite Dhruv to present his project. Over to you, Dhruv.
Thank you.
Dhruv, it would be great if you introduce yourself too before you start your presentation.
Okay. Is my screen visible?
Yes, it's visible.
So hello everyone, my name is Dhruv Mehra, and I'm a Bachelor of Science Honors in Medical Biotechnology. So last year I graduated from Amity University, Noida, and at present I'm working as a research fellow at Pine Biotech. And talking about my past experiences, I've worked as an intern in research and Sanger department of a precision medicine company. So there I used to work with my team on projects related to genomics. So it all revolved around NGS and DNA sequencing. Other than that, I've worked as an histopathology intern at a government hospital here in New Delhi, India. So talking about my research interests, so my research interest revolves around precision medicine, cancer biology, genomics and transcriptomics. And through this fellowship, I'm actually, developing an understanding of data science as well. So today I'll be presenting my progress of research, my research project called Transcriptomic Analysis of Esophageal Carcinoma. So I'll just quickly give a very brief introduction. So esophagus. Esophagus is actually a muscular organ that can interference to the stomach. Esophageal carcinoma is a malignant neoplasm of esophagus. So it has two predominant histological types called the esophageal squamous cell carcinoma and esophageal adenocarcinoma. So this disease is actually associated with tobacco use, alcohol consumption, and a deficiency of vitamin A, B3, and C. Along with that, the disease is actually related to various health conditions such as esophagitis, celiac disease, and GERD. So talking about the epidemiology, so these figures are actually from the WHO Global Cancer Repository. So these are figures from the 2020 data. So talking about the esophageal carcinoma, there were more than 600,000 cases of esophageal cancers, and the resulting mortality were more than 540,000. So these figures are quite high, and it indicates the need of new therapeutic diagnostics and prognostic interventions for treating this disease. And this whole idea is the reason why I'm undertaking this project. So before describing my hypothesis, I'll just describe my data set. So I'm using, for this particular project, I'm using the data from patient cohort TCGA. So the Cancer Genome Atlas, I'm using their esophageal carcinoma dataset. So this dataset actually consists of 198 samples, and so like 185 are tumor samples and rest are the normal samples. So here I would like to highlight these normal samples are nothing but the adjacent tissue to the tumors, and I've retrieved them from the TCGA database Firebrowse. So talking about the hypothesis, through this research project, I'll be working on two major objectives. So my first objective would be to know more about the gene expressions associated with outcomes in patient. So luckily I've been provided with a ton of metadata. So for carrying out this particular objective, I'm actually looking at the vital status. Since it is a patient cohort, so I'm provided with the patient status, whether the patient is decreased or whether the patient is alive. And using that, I'll be looking at gene signatures or precisely biomarkers associated with a prognosis of the disease. And my second objective would be to know more about the gene signatures involved in cancer progression. So for that, I'll be actually comparing the late stage esophageal carcinoma with the early stage esophageal carcinoma. So here is the analysis plan for carrying out my objective one. So I started by preparing my data set. So I started by matching my metadata with the expression data. Then I performed the differential gene expression. So I categorized my data into two groups, the decreased versus the alive. So as a result of that, I got 70 differentially expressed genes with a p adjusted value of less than 0.05 and log value of negative or positive 1.5 log fold change. So just to dive deeper into prognosis, I've actually plotted the Kaplan-Meier plot. So in this slide, there are actually two figures. So first, I'd like to describe the box plot on the right. So the box plot is about the expression of gene MDF1C. So this gene is one of the 70 genes which I have found via my differential gene expression analysis. So it is clearly evident that the MDF1C gene was downregulated in decreased group when compared with the alive group. And based on that, I've actually plotted a Kaplan-Meier plot. So talking about the Kaplan-Meier plot, it's basically a non-parametric test used to estimate the survival function. And this statistical analysis is usually done for knowing the survival analysis, for doing the survival analysis. And to simplify it, I've divided, for sake of explaining, I've divided into two parts. So first is a survival curve and second is a log rank test. So talking about the survival curve actually represents the survival probability over time. So talking about this graph, so there are actually two groups. Again, the low MDF1C group and the high MDF1C group. So talking about the survival curve of the low MDF1C group, it is clearly evident that it is, dropping significantly over time, while the high MDF1C group, in case of high MDF1C group, it is not the case. So, but just looking at the curves, we cannot say anything confidently. And then the log rank test comes into play. So the log rank test is actually a hypothesis test. So this actually works by setting up a null hypothesis that there is no difference between the survival function of the two groups. And in this case, there is no difference in survival function of high expression MDF1C group and the low expression MDF1C group. So the result of this log rank t-test was 0.033, which is less than 0.05. And as a result, we just, reject our null hypothesis, and we can confidently say that there is a difference between the survival function of these two groups. So based on this analysis, I've actually modified my analysis plan. So I'll be actually taking help of these. So I'll be actually performing a supervised machine learning random forest. So during time I was doing the transcriptomics module, I glanced upon this algorithm, and I still remember the special feature about it, that was the feature selection. So feature selection is a unique, it's a property of, not a property, but it's a criteria for distinguishing two groups. So these, so like first I'll proceed it by dividing my data into trained data and test data. So the algorithm will work on, will help me in finding those gene expressions which makes the difference between the alive and the decreased group. And I'll be presenting my progress next week. Thank you.
Just a question there, Drew. How many groups are you going to use for your classes? How many classes do you have?
Sir, only 2.
Yeah. So typically in classification, when it's just 2, it's very similar to differential gene expression, right? You're looking for genes that are differentially expressed, but differential gene expression shows you all of the genes, and classification tries to limit the number of genes. So it tries to select only the top features that contribute to accuracy and decrease in accuracy and precision. So if you have only 2 groups, I don't personally see the reason why you would go for classification as your main method. I mean, you can use it maybe downstream, but I would try to go back to your original plan and look a little bit closer at the metadata that you mentioned, that you have a lot of metadata about this, right? And think about, machine learning, the major challenge that it addresses is the high variability in the gene expression itself. Because when you do, let's find all the differentially expressed genes, like kind of what we saw in the previous project presentation. You see actually a lot of variation in the PCA. It's not a straightforward PCA. There are some outliers. There are some, groups that are closer or farther away from each other. And that's where machine learning is the best use for it. It looks for variation that is not straightforward, that is not clear. The pattern is a little bit more complex. And that's why you can combine then this kind of feature selection with clustering not of samples, but of genes, to kind of see whether you have this, co-expression of different sets of genes. So I think the overall kind of design is there. You've got the major pieces of the puzzle, but I would refine some of these steps in the way that you prepare the data.
Okay, sir. Okay, so I'll look into it, and I'll be presenting my results in the next week.
Okay, so thank you, Ilya. I have also some points. So I mean, I think especially for the machine learning, when I was talking about the tumor stage classification, because we have 4 stages, remember? So
yes,
that is, yeah. So at that point of time, I ask you, maybe you can apply supervised machine learning, because for the tumor stage classification, because we have 4 classes, then after identifying some important genes, then we can cluster early stage 1 and stage 2 samples as early, and stage 3 and stage 4 as late stage samples. So for that purpose, I mean, you can apply supervised machine learning, particularly when you have like already 4 tumor stages. Okay, so another thing that I just want to add, can you go back to your this Kaplan-Meier plot?
yes.
So what I was looking at, like you have total samples, if I do remember, nearly 180. But if I'm seeing, looking at this, you have high risk and the low risk samples only 46 and 46. So whether there is no survival information is available for rest of the samples, or what is this? And another thing, like you are talking about low MD FIC value. So are you taking specific only one gene, right? Why
Yes,
you have taken only one gene? So I mean, especially Kaplan, this overall survival analysis, or survival analysis specifically helpful once you have already identified some important genes under some conditions. For example, you have, you are trying to identify early or the late stage samples, and you have identified these are the important genes. So then you can go for this type of analysis. Maybe they are playing important role in the prognosis. So why you looking at one gene? So I am my first question was about that, and the major question about these groups, because I just want to know, like, whether you don't have the survival information regarding all the samples, or exactly what was the, why it is like 46 and 46. It's just like 92, total 92 samples.
Okay, ma'am. So, ma'am, I have plotted this using a tool called GPR. So that also uses the same TCGA data as I'm using. So I guess the probable reason would be the non-availability of that data.
You have to explore that. I mean, whenever you're using particular tool, you must understand that. Because you should analyze, like, totals, you have total samples this much, so why you are getting only these groups, right? And in fact, this is again a significant, when you are actually interpreting these results, you are saying like this is not significant or something, but because you have already p value is significant. But apart from that, you are also get the HR value, that is hazard ratio. So
Yes,
you also got that value, so that will let you know whether it is actually good prognostic factors or the bad prognostic factor, depending upon whether it's positive HR value or negative HR value. So that is what I think, that would be after you have already analyzed, and then you must go for the prognostic analysis. I think it should not be your first part. I mean, then when you are actually doing the analysis and identify some important genes, maybe between early or the late stage, then maybe you can do this analysis for those genes.
Okay, ma'am.
Yeah. Okay.
Okay. So I'll be working on the points, and I'll be sharing my progress next week.
Yeah. Before even that, at least, I mean, go for the PCA analysis. At least that will help you to understand whether these classes able to differentiate or not. So then we are identifying some important genes that may be able to differentiate these early or the late stage samples. Yeah.
Okay, ma'am.
Okay.
Thank you so much, Dhruv, for sharing your research with us. Now, I would want Peniel to share his research progress. Over to you, Peniel.
Okay, thank you. Just a minute, let me share my screen.
We can see your screen now. Thank you.
All right. Hello to everyone. My name is Peniel. I am presently in China, but I am a Tanzanian by nationality. I am doing PhD here at Suzhou Medical University in China, and my research interest is neurodegenerative diseases. Particularly, we are into Parkinson's disease and glioblastoma. That is what our lab deals with. So for the sake of this presentation, for the sake of this research fellowship, I'll be talking about Parkinson's disease, and for this case, I've come with a topic, which is the novel biomarkers of levodopa-induced dyskinesia in Parkinson's disease, a potential target for anti-dyskinetic therapy development. So PD is actually the second most common neurodegenerative disease that affects 2 to 3% of the old population, and it is estimated that this might increase in the future. So basically, what it is observed in clinics is some symptoms, and the 4 commonest ones are rigidity, the tremors, and bradykinesia. And of course, it started with Parkinson himself in the year 1817. And what is actually the pathophysiology of it is the loss of dopamine in some nuclei that are responsible to produce dopamine. And we have some nuclei in the brain, and for the sake of this presentation, it's good for me to highlight because that is the metadata talks about. So we have this nuclei over here, we call it striatum, and we have globus pallidus and substantia nigra. So there is 2 pathways that are involved in dyskinesia or movement regulation. There are 2 pathways because I'm talking about the 2 pathways because the metadata I have is talking about those 2 pathways. So we have one pathway here which starts from striatum, these 2 nuclei to this point. This is called striatum pallidus, and here we have striatum nigro. So striatum nigro is a direct pathway, and striatum pallidus is indirect pathway. So direct pathway, in a nutshell, activate the movement, and indirect pathway stop the movement. So if you have loss of dopamine, and these 2, all these nuclei that are responsible to produce dopamine, definitely the motor system will be affected. So at the end of the day, the person will present with abnormal movements. So in the provision of integrated treatment for patients with this disorder, the discovery of dopamine as a neurotransmitter in the brain and deficiency in PD has been, and levodopa as a therapy has been the major revolutionary event. But the main problem of levodopa, it causes what we call dyskinesia. So it is reported that overuse, chronic use, or high dose of levodopa use, it will definitely cause a dyskinesia. And this is how the dyskinetic patients will be looking like. Abnormal movement that can affect the lower limbs or even the upper limbs. So the development of the new therapeutic interventions to mitigate the risk of dyskinesia and PD, that's a high priority. So to uncover this, we have performed a bioinformatics analysis of microarray data set GSC 55096 from a geo database to identify differential expressed genes using Lima package, gene ontology and KeyG, and later also talk about the protein-protein interaction will be used to describe those differential expressed genes. So just in a nutshell, this metadata contains 77 samples, and it has got the 2 pathway, direct pathway and indirect pathway, as I mentioned earlier. And this direct pathway, and these are the model. It's a model of direct pathway and a model of indirect pathway. And this model, they are treated with Parkinson's disease toxin. This is a toxin for Parkinson's disease, and the control as well, similarly to indirect pathway. So in Parkinson's disease model, later on, though, they treated with chronic high levodopa, chronic low levodopa, and saline. Similarly to indirect pathway, but for only Parkinson's disease, they treated with chronic high levodopa, chronic low levodopa, and chronic saline. So we have discovered that chronic high levodopa versus chronic low levodopa phenomena, levodopa treatment reveals no significant molecular changes on indirect pathway, but rather on direct pathway. So we checked. So what we found in chronic versus chronic high versus chronic low on indirect pathway, actually with UMAP, there was no much separation of these two dosage when we were talking about chronic low and chronic high levodopa. And with volcano plot, we could not get more upregulated, neither downregulated genes in this indirect pathway. This could indicate that this indirect pathway, the molecular changes on this indirect pathway are not as much as it is in direct pathway. So the only gene that we actually found is one gene, and is higher in low levodopa compared to chronic high levodopa treatment. So in a direct pathway, we found that there is a clear difference between the high dose and the low dose of levodopa in UMAP, and with a number of upregulated and downregulated genes, actually up on 90, 87 upregulated genes, 80, 27 downregulated genes were observed in this direct pathway. So this is just to represent a number of genes that and how it looks like in these two dosage, in chronic high and chronic low. And for example, this gene, it is higher in chronic high levodopa than in chronic low levodopa. So we actually took 20 upregulated genes, and so as to visualize their molecular, biological, and cellular and the pathway. And for molecular, for example, we found that most of those genes, they are involved in binding, in binding, and for biological processes, most of those genes are involved in cellular processes and metabolic processes. And similarly to cellular component, we found that most of the genes were involved in cellular anatomical entity. And for the pathway, most of the involved pathway in those upregulated genes is TGF beta signaling pathway. So now, if one is talking about to mitigate or to attenuate dyskinesia, we thought of using the upregulated genes. If one is thinking about the drug discovered to attenuate the dyskinesia, we used we checked on the upregulated genes, and for this case we used the direct pathway, because the direct pathway seemed to have more and clear separation of the dose. Then this is, I mean, the high dose, low dose, and the control, which was saline. And we found 1,049 genes that were intersecting between these three groups. And we checked on the volcano plot, and we found this is how the volcano plot looks for chronic low levodopa versus saline, and chronic high levodopa versus chronic low levodopa. And this is for chronic high levodopa versus chronic saline. Saline was just a control group. So we took, again, top 20 upregulated genes, and most of these genes are, again, they are very important because we also deal on these genes in some aspects. For example, NUR4A3, this is a transcription factor, and we also deal with this gene in our lab. And most of these upregulated genes are involved in binding, and most of them are transcription factors. But we checked on GO and KEGG, and we found that most of these genes are also involved in binding. And for biological processes, most of them are they are similar to the rest of the other genes that we checked earlier. For example, in biological processes, most of them are involved in biological regulation, cellular processes, and metabolic processes. So for the cellular component, most of genes are also involved. They are involved in cellular anatomical entity. And for the pathway, most of those upregulated genes are involved in CCKR signaling map. So, and for the protein class, we found that most of these those genes, they are involved in gene-specific transcriptional regulator. They are gene-specific transcriptional regulator. So this is what we will do next, because at the end of the day, we have to validate those genes, and perhaps if it can later be used as for treatment, perhaps maybe to use inhibitor of these genes in order to regulate or mitigate the dyskinetic movement that are appearing to this Parkinson's disease models or humans later. So we have selected a number of genes according to the log change and the t-test. Adjusted p-value and their log change. And these are the genes that we are hoping to validate. And what we have to do is to make the Parkinson's disease model. We are making those models over here. It's not a big problem, and we'll later do the behavioral test of those models and Western blotting. Again, we have to, during confirmation of these genes, we have to do qPCR, Western blotting, and immunohistochemistry. Thank you. This is what I plan to share for today. Thank you.
Thank you, Piniel. I think it's a very interesting project. I must say that I have quite a number of questions about how you kind of started your presentation today, and what is the plan overall. So I wonder if we can schedule a separate meeting. Maybe Sonalika and Dr. Harpreet can help us coordinate, because today's session, we already have a pretty long session already. But I would love to learn a little bit more about the data set and the different treatment objectives that you have. So thanks for the presentation though.
Sure. Thank you very much.
Thank you so much, Peniel. So we have many new research fellows from Amity and Punjab University as well, and they have introduced themselves to our mentors. So I won't be asking them to introduce again, but we have Asha today. So Asha, can you please introduce yourself? She's a PhD fellow. I request you to please keep your introduction short because we're running out of time. And we'll then schedule a meeting separately with you and take your research interest there then. Over to you, Asha.
Hello, everybody. Myself, Asha Abraham, doing PhD in SRM University, India. Myself from computer science background. I'm in the first year. I like to do PhD in to predict a deep learning to prepare a deep learning system, to propose an algorithm for transcriptomic by making use of the transcriptomic analysis of ovarian cancer. That is prediction system for the biomarkers of ovarian cancer by linking with precision medicine.
Thank you so much, Asha. So for all the research fellows here, we keep on having trainings, monthly trainings on different sorts of topics. So we have this training coming up on machine learning for biomedical drug discovery, and then we have chem informatics for biomedical drug discovery. And all those who are a part of a research fellowship program, they would get the access to be a part of the trainings as well. So I would be sharing the details with you, and then you can join the training too. And also you can be a part of our ongoing trainings. I keep on sharing the links with you already. So it's like those who are interested in different sorts of research areas, they can pick up whatever training they would want to go ahead with, right? And now I would want Sabita to introduce herself. Over to you, Sabita.
Thank you, Sonalika. Hello, everyone. My name is Savita Tamang, and I'm from Nepal. I have graduated with BTech in Biotechnology degree from Purvanchal University, Nepal. So currently I'm working at different labs in Nepal. I'm working as a plant tissue culturist at a private biotech company. And I'm also working at a Center for Analytical Chemistry at Kathmandu Institute of Applied Science. There I'm looking at the prevalence of low quality drugs in Nepal. So the reason why I joined this research fellowship program is, as we know, biotechnological developments are increasingly dependent on the extensive use of data generated by high throughput instrumentation. So through this program, I believe to learn from experts about data integration, exploitation, and exploration so that I can implement them in different biotechnological areas. Thank you.
Thank you so much, Sabita. And now I would like to tell you all something. So we have had a lot of enrollments in the research fellowship program. So we are planning to make up batches of these research fellows, and then we would take your progress there. So all the Amity students, the Punjab University students, and other students as well, we could not get to your progress, but yes, we have gone through the document which you filled out. So those who have filled out the weekly research fellowship program form, good. And those who have not, please fill it out. We would want to know where you are in your weekly progress, right? And next week when we meet, I would want other research fellows to please prepare 2-3 slides and share where they are in this program right now and what is their progress. So say if you have completed transcriptomics part, like the course transcriptomics part 1, 2, 3, then please share what you learned in those particular courses, and then we can take your queries as well. And those who have completed the courses, then please fill out the research proposal form, and then we will discuss what your research proposal should be and what your modifications in the proposal have to be, right? And now I would want to hand over to Dr. Harpreet if she wants to say something. Over to you, Dr. Harpreet.
Thank you, Sonalika. So I just want to inform everyone, please, whenever you have one-to-one meeting, before that, you must fill that progress form. Okay? That is like compulsory for everyone, whether you are completing courses or whether you're completing project or even if you have done something. So you have to fill that form, and on every Monday we used to share that form with you, and please fill that form on Monday itself so that we can look into with whom we need a one-to-one meeting. So that's it, and the important, that is like compulsory for everyone. It's not like your choice, and then only after I get the response from there, and then we are going to schedule one-on-one meeting. Yeah. So thank you everyone for presenting your work, and I'm looking forward from others who are just starting the courses, so that once you're able to complete the courses, then we will going to work on your own project. So thank you.
Thank you so much, Dr. Harpreet. And at last, I would want you all to also accept my invitation of the Slack channel, because it's really very easy to communicate things directly there, right? And those who have not received the link yet, I'll share with them once again, and then you can proceed further. Thank you so much, everyone, for being a part of a research fellowship meeting today. And if you have any doubts, so we had a number of participants who are going to enroll themselves for the program. If you have any doubts, then please mail me at marketing@pine.bio, and I'll be there to assist you with your enrollment. Thank you so much, everyone. Have a great day.